It is time for us to address the remaining part of muscle tissue, which has not been introduced in a previous video presentation. Today, we're not going to spend too much time reviewing what has been already discussed about the structure and composition of either skeletal or cardiac or smooth muscle fiber. Rather, I'm using just those few diagrams that were used before to put us back where we stopped and to allow us to continue without any interruption into sections that were not discussed so far, which predominantly for this presentation is contraction of the muscle, aka sliding filament theory. So here we are at the beginning, just to try to refresh our memories about how one skeletal muscle is composed. It is made of individual muscle fibers that are thrown together and bundled into fascicles or bundles, and then multiple bundles would be involved in structuring one peripheral skeletal muscle. Individual endomysium, perimysium, and epimysium, connective tissue membranes surrounding individual fibers or fascicles or covering the entire muscle, are those ones that continue past the boundaries of the longest muscle fibers or muscle cells. They become all bonded together, integrated into tendon of a muscle, for which we said it could be either a tendon of origin, muscle being attached to a stationary part of the skeleton, and with its other end showing tendon of insertion, which is observed on a bone or part of the body, which moves when that muscle is contracting. So origin, insertion, stationary versus mobile part of the body. Individually, single muscle cell would be much more of our concern to continue our studies, because when we open its membrane, the sarcolemma, we see that inside a cell, which is quite elongated, hence the term muscle fiber would be more appropriate than just to call it a muscle cell. We're seeing lots of different myofibrils, also structures that look like some kind of very small but parallel running cylinders going from one end of the cell to the opposite side. And of course, the individual myofibril, when further analyzed and broken down, would be shown to be made on the basis of multiple repetitive segments that are simply called the sarcomeres. On the surface of individual myofibril, we're going to see the cause for this kind of light and dark alternating pattern on the surface of a cell when observed under the light microscope. We said that it is due to arrangement of contractile proteins within the muscle fiber, which is quite tightly regulated, and that's why it is not a random allocation of light and dark bands, but rather something that follows a very specific rule, and we're going to see it quite shortly. So for us, for now, it's just important to understand that contractile proteins inside striated muscle cell are practically arranged in structures recognized as myofibril. And each of the myofibril would be made of a smaller subunit, which we're going to observe shortly and understand what arrangement of contractile proteins really looks like. This picture we also introduced, and it's going to come handy towards the end of this presentation. This is a neuromuscular junction. We see here axon with its terminal ends, and some kind of synaptic end bulbs could be also noted here, where underneath the nerve, we see three different skeletal or straight muscle fibers. We can see those alternating light and dark bands. But this photograph, magnification of 1,650 times, will be actually important for us to understand how the nervous signal action potential comes from nerve onto the muscle and stimulates the muscle to do contraction. So although this diagram also had been in use before, we are not focusing any longer on what resulted in appearance of exceptionally long muscle fibers, cells that are characteristically recognized with several nuclei within the cell, but rather we're going to spend some time talking about much smaller structure that is called the sarcomere. On the next slide, you're going to see maybe a little bit more of what defines the sarcomere. We can say it is part of the same myofibril, which is observed between two consecutive Z discs or Z lines. That is the length of a sarcomere. While we're still at this diagram, we can see that some kind of thinner and a little darker versus thicker and brighter red-colored lines are added onto this diagram. They represent those contractile proteins, and the M line is actually very middle of a sarcomere. So we're going to come to this diagram shortly, but before we do that, let's try to understand what else is important for us in order to understand this part of physiology. So here is a myofibril, and here's another one pulled out from a cell, but being cut in half so we can observe, oh yes, truly, it is composed of sarcomeres, and we can see this zigzag line that is the Z disc, and here's another Z disc here, so we know that distance from this point to this one actually represents the length of a sarcomere. Term sarx, I believe I introduced, actually is the Greek word for flesh, for meat, and that's why a number of different structures within this segment of physiology do have a prefix sarx, such as sarcolemma. We do not necessarily call this cytoplasmic membrane, we simply use a new word saying this is sarcolemma. Inside a muscle cell, we would not be talking about cytoplasm, we would be talking about sarcoplasm, which very clearly points out to, okay, well, it is certainly happening within a muscle cell if the prefix sarx is used. What do we have here in the vicinity of individual myofibril? You see here a set of very irregularly looking yellow-colored tubular structures that appear to be quite large in the vicinity of Z disc. And then as it goes towards center of the sarcomere, towards the midline, it becomes a little bit more irregular, composed of tubular spaces, a variety of different shapes and diameters. And then as we're coming back to another Z disc area, this appears to kind of just repeat itself as a pattern. We have also several mitochondria that are part of this diagram, but they're not necessarily in our focus. So what we have just added to this diagram, and hopefully added to our knowledge, is what is called the sarcoplasmic reticulum. It will remind you of endoplasmic reticulum, but this is muscle tissue. And instead of having a role as allocated in a cytology section, the sarcoplasmic reticulum would be actually amazingly massive source of ionic calcium, which is an ion without which muscular contraction would not happen. It has to be released from a sarcoplasmic reticulum, and a release cannot be done on the basis of some kind of wireless connection or Bluetooth connection. It has to be a little bit more analog. So we can see here that when we're talking about sarcoplasmic reticulum, its most peripheral, much wider sections are called terminal cisterns. And there are two of them next to each other, but in between two terminal cisterns, we have a structure that is called the T-tubule. Now, in this part of the diagram, although it's not labeled, I can point what T-tubule is and try to bring it a little bit closer to your perception of what is about to happen. Obviously, the T-tubule and two terminal cisterns of the sarcoplasmic reticulum, they form a triad. And it is not possible to say which one is more, which one is less important, but this is the situation which I would try to describe for you, but just using a little bit of a different perspective. This is the sarcoplasm, and then it's massively indented part that goes deep into the cell. This one, we can refer to it as the T-tubule. This is surface, and this is indentation of sarcoplasmic membrane deep into the direction of the cell. Next to its end, we said we're going to have to place two different terminal cisterns containing calcium ion. Here is first one, here is the second one. So this area here within this box, that is what diagram below shows and points as the triad. If the endoplasmic reticulum contains calcium, it needs to know why and when to release it. And certainly it won't be a spontaneous event or event which is done randomly. Instead, it is going to be like this. When a nerve cell delivers its action potential on the surface of a muscle cell at the neuromuscular junction, it immediately starts affecting motor end plate of the cell, this segment, to start changing polarity of the sarcoplasmic membrane. So as the polarity changes, I will just put here a couple of plus signs just to identify, that is the electricity that keeps now traveling from neuromuscular junction over the rest of the muscle surface. Charges also go deep into the body of the cell using T-tubules. Otherwise, a cell, as per laws of physics, would always remain like a Faraday's cage. Electrical charge applied to it stays on the surface of a cage and nothing changes inside the cage. That's why airplanes are considered to be a good example. Airplane could be hit by lightning. However, nothing is going to happen to passengers or crew inside the plane because the electrical charge actually will dissipate over the surface of a plane without chance of getting into it past the shell. So the T-tubule essentially just allows as a conductor for electrical charge to be diverted deeper into cellular structure. And at this point here and here, it will start affecting endoplasmic reticulum, cisterns, terminal cisterns in particular, from which massive release of calcium will be observed in order to stimulate interaction between proteins, contractile proteins of this cell, to start seeing further interaction, resulting in a shortening of a single sarcomere, but in a big picture, shortening of the entire myofibril, which translates into shortening of the muscle between its tendon of origin and tendon of insertion. So at this point, we just need to confirm. New interesting thing for us is addition of triad, two terminal cisterns of endoplasmic reticulum containing plenty of calcium ions are put together with a T-tubule, the indentation of the cellular membrane, allowing access to electrical charges to get deeper into the direction of the cell to avoid the example of Faraday's cage. Now, when we know practically what's going to happen, electrical signal arrives at the surface of a muscle, T-tubule, a release of calcium, calcium required for contraction to happen. We now need to explain this step by step and to really try to understand a little bit better what is the structure of individual sarcomere, which is the base building block of a myofibril. We already defined it as saying like the length of a sarcomere is the distance between two Z discs, those irregular zigzag lines. And you can see here on the lower part of the screen what the Z disc is. Actually, it's also protein, one of the structural proteins within the muscle, which of course is illustrated in the form of zigzag line, but it's not a bone or a cartilage or any other material. It's also protein. So as we noticed, left and right side of sarcomere is defined by the distance between two consecutive Z discs. In the middle of the sarcomere, we still have another protein that will help us regulate stuff inside, and it is what shows as another thickening, not too wide, but rather just being called the M line. Okay, so with addition of M lines and Z discs, you and I now have more space to talk about proteins that would definitely do the work, and we call them thin and thick filaments. It's a little bit of a euphemism because they're proteins which have their names, but the way we see them is one looks thinner, one looks thicker, which you can also get the idea on the upper part of the screen which one is which. And you realize that thin and thick filaments do have the area where they overlap each other. And then there is part of sarcomere where thin filaments do not overlap thick filaments, and there is part where thick filaments are not overlapped by any of the thin filaments, which again points very directly that this part from Z line to here actually is reflective of this fairly light bend of the sarcomere. Segment of the sarcomere where thick and thin filaments are heavily overlapping each other is this segment of the sarcomere. And then from this line to M line, we have another part where thick filaments exist, but they are not overlapped by the thin filament proteins. Same thing would be seen also on the other side of the M line, so no need for us to go and duplicate the work. Area where just thick filaments exist without being overlapped by thin filament fibers is known as the H zone. And then to the left and right from the H zone, we have the zone of overlap. And it is quite stationary to observe it like this, because if we would consider this being carved in stone, practically we would get stuck with length of a sarcomere being always the same. But when we add a little bit of dynamics, we can see here that zone of overlaps, two of them, and H zone essentially represent the A bend part of the sarcomere. I bend goes from the end of the thick filaments, not to the Z disc, but to the end or to the right side of the next sarcomere, where we start again seeing appearance of thick filaments, which would be about here. That's the I bend. So where is that dynamics that I was talking about? Very clearly, we can start thinking that between thin and thick filaments, yellow and red colored ones, interaction could be forced to change, and that there would be more significant increase in the overlap zone, whereas interaction between the thin and thick filament is what we see and describe as the sliding filament theory. So we can see eventually shortening of a sarcomere, bringing this Z disc to the right side, while this Z disc becomes closer or being moved to the left. Obviously, it will result in the shortening of the sarcomere's length, more intense overlap between thick and thin filaments, but that is part of the process that we're trying to explain and to go with some additional details. Anyways, with the idea of proteins, thick, thin filaments, proteins acting as the M line or Z disc in place, zone of overlap, and zone which is H zone, no overlap, you and I can move safely to the next slide and to start looking like what exactly those thick and thin filaments are made of. Answer is relatively simple, but we have to use a couple of extra words to make sure that description matches what's happening in a natural form. Obviously, the thick filament is made of myosin molecules. Myosin is a protein, which, as you can see, looks like two golf clubs being braided through their handles, but only their heads appear to be separate. So this forms a myosin tail, and the myosin tail is embedded into the core part of the thick filament. Myosin heads are those rounded projections that are sticking out. They're making thick filament looking even wider as some kind of projections, and they're called the myosin heads. We're talking about 200 plus myosin molecules being thrown together to assemble a single thick filament. On the other side, the thin filament, which again, due to illustration here, appears to be thicker than the one which is on the top, but please disregard this disproportion. We're going to have thin filament being a little bit more complex in its structure. It is mostly going to be made of these yellow-colored molecules of actin, so myosin actin. And actin molecules will have to be attached on another supporting molecule that is called the tropomyosin. So it may remind you of grapes, where multiple individual structures in the form of little sweet, juicy bubbles are attached on the same stem. So if you're using that comparison, it is okay. So molecules of actin are attached onto molecule of tropomyosin. But you must notice this kind of darkened structure that exists on individual molecules of actin. These darkened areas are representing the myosin binding site. Now I'm telling you about 1/3 of the story. Obviously, thin and thick filaments will have to interact with each other. How is this going to happen? Answer is relatively simple. Myosin heads are going to bind onto myosin binding site of actin molecules, that kind of dark spot. But now you can see that on this diagram representing just a thin filament, a part of it, myosin binding site is covered by another protein, which is called troponin. So for as long as we have situation like this, status quo, myosin binding site of actin will not be available. It won't bond with anything. It'll stay hidden. So first thing is that we need to do something to troponin. And when we do that, it is going to allow myosin binding sites to be uncovered, to start showing in this position, or in this position, or in this position, so that they become visible and accessible. Therefore, we're expecting then interaction between myosin heads with actin molecules, but on a very specific location on the myosin binding site. Actin attached to tropomyosin, and then troponin, the molecule which prevents or covers myosin binding sites of actin molecules. If this is not too much, we can now see what's happening potentially when we observe two consecutive sarcomeres. Here's one from Z disc to another Z disc, and here's second one. Obviously, they have H zone, I band, zone of overlap. A band includes the zone of overlap on both sides, and an M line. But keep in mind, this is relaxed muscle. As the muscle is partially or maximally contracting on diagrams B and C. You can see it. Obviously, area that is overlapping thin and thick filaments essentially progresses. We have more overlap on the partially contracted and definitely like the maximal overlap. It could be observed just simply by measuring the distance between two Z lines. And that is why we call this the sliding filament theory. Obviously, thick and thin filaments, just like my fingers, if you don't mind taking a look at small picture, I'm just allowing fingers to slide across each other and to just have a bare touch at fingertips. So this is relaxed, and this is the sarcomere, which goes towards its maximal contraction. Now, we just have to explain what is the mechanism that leads into this set of events. We understand what it is, but we need to be able to explain what are events, what are causes, what are consequences. And we're going to use this diagram, which really is fairly simple, fairly straightforward to understand and to practically start putting pieces of sliding filaments theory together and make it one nice rounded unit. So the first thing that happens is that the muscle must have the energy. For muscular contraction, as we know, energy is a must because it's a process that will require that we overcome some natural resistance, such as gravitational force, or to do a number of different things where our body will be going against our own proverbial gradient. So we will have to either climb the hill, run a flight of stairs, carry the load, which requires muscular contraction, therefore consumption of energy. It really scales down to molecule of adenosine triphosphate, which binds to the myosin head and immediately becomes hydrolyzed into adenosine diphosphate and a phosphate group, which is released into sarcoplasm of a cell. As you can see, we have actin molecules and the myosin binding sites revealed compared to the previous diagram. It is troponin molecule, the one which is having dark blue grayish color, which accepted these two little purple bubbles on the top of it. That is calcium. So calcium would be another thing required, and we have that information in the center of this diagram. Contraction cycle continues if ATP is available and calcium, ionic calcium, is present. So calcium is released from endoplasmic reticulum, double positive charge for calcium ions. It binds onto a molecule of troponin. And now troponin practically allows actin to kind of rotate, just change their position relative to a molecule of tropomyosin and expose their myosin binding sites, these darker spots that you can see on practically each and every yellow colored actin molecule. So that is the moment that we were waiting. We know that T-tubule and sarcoplasmic reticulum would be stimulated to release the calcium. Muscle fiber will consume some of its previously stored adenosine triphosphate. And as a result of this, on the diagram number 2, we see what's going on. It is the myosin head which binds to actin on the myosin binding site. And the formation of this temporary bond between thick and thin filaments is what we say it was forming the cross bridges. I forgot to say, but I believe that you can see it, that molecule of myosin has some kind of hinge-like mechanism allowing individual myosin heads to move away from the core of thick filament in order to make the cross bridges with actin molecules. So when the cross bridges are formed, adenosine diphosphate is still present, and we know that it is going to result in further breakdown, that we need to use more energy, as diagram number 3 tells us that now the myosin heads will rotate towards the center of the sarcomere, the M line. So you realize that this is something as equivalent as if you hold yourself onto a rope. So you are temporarily the thick filament. With your hand, you attach to rope the thin filament, and that rope is going to be used to allow you to safely pull your body through a swimming pool practically to its end. So what you will be doing with your arm, you would be grabbing a rope and just pulling it, but because it's attached to the wall, you can't pull the thin filament rope towards you. Instead, you're holding onto attached thin filament, and you are allowing your body to keep moving relative to a rope. If there's need for it, I would just go quickly back and confirm. You see the thin filaments are attached physically to the Z disc. So when you hold to them and you try to pull, basically you are not pulling them individually. You are just pulling thin and thick filaments into more significant overlap. So it will look, when you observe the entire sarcomere, that with a contraction of the muscle, both ends, Z lines, are pulled closer to the M line, the middle point of sarcomere. So this analogy that I was using, that you are holding on to a rope which is attached to the pool, is just to make sure that you understand your grip on the rope is temporary. So when you hold on to it and you pull, your body moves relative to the rope. But in order to continue movement, you need to let rope go out of your hand, grab another segment, and then again invest the energy in order to produce another pull attempt and complete the action that you started. So the pull moment is actually, in sliding filament theory, described as the power stroke. Myosin heads rotate towards the center of a sarcomere, producing movement which occurs between thick and thin filaments, and that is the power stroke. So finally, diagram number 4 is what I just tried to explain. In order to pull yourself through the swimming pool, you have to let go the rope out of your hand, reattach it to a different one, pull, reattach again, and do it several times. So as the power stroke happens, basically just a minimal movement between thick and thin filaments happened. But now the myosin head, which is still energized, could form another cross bridge. And as a result of that second cross bridge being formed, different part of the thin filament is held and different power stroke could be applied. So the sarcomere goes into maximal contraction. Finally, when it's done and the contraction is practically reaching its peak, its maximum, it's time to allow muscle fiber or sarcomere or an entire muscle to relax. And as a relaxation is just about to happen, we would need to detach cross bridges, myosin from actin, as it would happen at the end of contraction cycle, and muscle will try to re-energize itself and to be ready for another event of the same kind whenever it is required. So in one short term, it doesn't appear to be excessively complicated. Now we have two comparative diagrams showing muscle at state when it's fully relaxed and also when the muscle is contracting. So here are our beloved T-tubules, and we know T-tubules will affect terminal cisterns, and they would use, as you can see, calcium active transport pumps, that is channel, which allows calcium to be freely manipulated. Being released is okay, but now being reuptaken back into terminal cistern obviously is requiring energy, because we'll try to store the calcium and to force it to move backwards into terminal cistern, practically against its own concentration gradient. Calcium channels at this stage are closed. Calcium is quite stationary, and we see here thick and thin filaments are in their given position. They do overlap, but there are no cross bridges. And the reason for that is very simple. Those myosin binding sites on the molecules of actin are hidden. Therefore, troponin is keeping them hidden, preventing myosin from sticking onto it. However, when we receive the action potential from a nerve, and it will generate some electrical changes to the sarcoplasmic membrane, electrical charge will dip into T-tubules. From T-tubules, it will affect terminal cisterns of endoplasmic reticulum. There will be a release of calcium into cytoplasm of a muscle cell. The calcium ions bind onto molecule of troponin. See the difference here on the left side without calcium, and now with two little purple calcium ions being added. It is the molecule of troponin which revealed the myosin binding sites, and the myosin molecules are forming cross bridges. So the next step, as we know, based on the previous slide, would be after cross bridges are formed, the power stroke is the one which continues. You can think of it both ways, either as, pulling a rope, or eventually power stroke if you are, let's say, competing in athletics and your favorite is javelin. So you will be holding it. So that's your cross bridge that you have formed. And then the moment you throw it and release it out of your hand, that's going to be a power stroke. So that is what's happening in contraction. Troponin holding tropomyosin in position to block the myosin binding sites. But when the calcium binds to troponin, it changes the shape of troponin-tropomyosin complex, and it uncovers myosin binding sites of actin. That is that kind of like scenery or stage that we have to set in order to ensure that cross bridges could be formed, just like north and south pole of magnets will stick to each other. That is that kind of natural attraction between actin and myosin, especially when binding site is revealed. Forming cross bridges happens very quickly. And then adenosine triphosphate, which has been already converted into adenosine diphosphate, is the energy needed to produce the power stroke. And as such, you and I are observing what the sliding filament theory is. Certainly, nervous and the muscle tissue must be integrated, because both tissues obviously use the similar language of electrical current and changes in the membrane polarity that eventually would affect each of the two cells in order to integrate them into one single functional unit that we call the neuromuscular junction. Upper left corner of the screen shows us one axon with its axon terminal, synaptic end bulb, practically being against the sarcolemma of a muscle cell, but still between these two, we do not have a direct physical contact. It's not like a nerve is plugging itself into a muscle, but rather the nerve with its synaptic end bulb actually adheres onto the muscle surface. It's equivalent of what we have described earlier as the synaptic cleft. We have the same little gap, except it's not between two neurons, it is between a neuron and the muscle cell. So what we need idea that we're talking about something very comparable to a synapse. Instead of two neurons, we have neuron and a muscle fiber. So we call it, for practical reason, the neuromuscular junction. Only part of the sarcolemma is good for that. Not this segment, for example. It would not be good. It is a segment where we already have here synaptic end bulb and part of the sarcolemma, that one which responds positively to changes induced by single nerve fiber or synaptic end bulb, is what we call the motor end plate. So here's a nerve, axon terminal, electrical current traveling down to the synaptic end bulb. It contains vesicles storing neurotransmitter, and they're just waiting for release. So whether we're gonna need more calcium channels to stimulate the release of transportation vesicles and to release the transmitting substance, it is not really so relevant. We can say it functions in the same form, because only with the electrical current changing the synaptic end bulb, movement of vesicles and release of neurotransmitting substance will be seen. In this case, it is substance which is called the acetylcholine, ACH. It is released from the axon, and acetylcholine molecules, just like any other neurotransmitter, travel down the concentration gradient from high to low, from nerve onto a muscle cell through the synaptic cleft. On the other side, motor end plate of a muscle already has proteins, protein channels that are actually ligand-gated channels. In this case, the ligand is molecule of acetylcholine. Those little red-colored three-sided pyramids are graphically illustrating what's going on. So when the acetylcholine binds to its receptor at the proteins of the motor end plate, it essentially opens up the channel to become more permeable. And again, the same culprit, the sodium ions, they keep moving very quickly into sarcoplasm. So practically, it's the same process of this part of the membrane, the motor end plate, will become depolarized. Its polarity will change. It'll become more positively charged inside. And of course, this kind of change will lead to muscle action potential being produced, affecting T-tubules, affecting, of course, sarcoplasmic reticulum, releasing calcium, calcium bonding to troponin, tropomyosin, revealing actin binding sites, and so on and so on. So we're going obviously back into sliding filament theory and the cycle that was previously described. What happens to acetylcholine? Well, it is something that I was waiting until this point so we can go through both nervous system synapse and also through the neuromuscular junction to address this as something fairly important. Acetylcholine is a neurotransmitting substance, and certainly it could be removed from this area if it further diffuses away, because we know this is high, this is low concentration, but somewhere else nearby is the area filled up with fluid with even lowest concentration of ACH molecules. So diffusion is one part of the process which prevents that once the neurotransmitting molecule was released, its effect becomes permanent. Nothing lasts forever. So muscular contraction in particular should be somewhat short-lasting, allowing muscle to relax, to get ready, and to repeat the action as many times as it's needed. So diffusion of neurotransmitter becomes one. Enzymatic degradation, or ACH broken down, practically means that ACH splits into a acetyl group and the choline group. So it's not ACH any longer, it's two different components. And of course, as such, they don't have the same capacity. They've been broken down and brought to the level of being inactive. Therefore, their action at the receptor practically comes to abrupt stop. And possible third way of getting rid of any surplus or excessive form of neurotransmitter is to consider it being taken back into a cell which has released it. So that is uptake by cell. And as such, we realize that it is the event which has its start, its mid part, and its conclusive part, beginning and the end, and is certainly facilitated by removing of neurotransmitter because its excessively long presence in a neuromuscular junction will trigger prolonged muscular contraction and, of course, faster fatiguing of the muscle, which is not necessarily our benefit. On this diagram, we can now sum it up all together, but essentially, we already covered this together in the past few minutes of this presentation. So it is really like including viewing or getting the idea of what's happening at the neuromuscular junction, nerve impulse arriving at the end. It is the axon, synaptic end bulb, which releases vesicles containing acetylcholine. Acetylcholine diffuses onto receptors which bind it, forcing more sodium to come into the muscle. And then at one point, acetylcholinesterase that exists as the enzyme within the synaptic cleft is removing or breaking down acetylcholine into acetyl and a choline group and practically inactivates it. In the meantime, action potential that was generated by this particular muscle on the basis of kind of localized change in the polarity of the membrane here travels down, it goes even into T-tubules, from T-tubules into sarcoplasmic reticulum, opening up calcium channels and allowing calcium to move from endoplasmic or sarcoplasmic reticulum into sarcoplasm. At this point, calcium, as we know, binds to troponin. Troponin then reveals actin binding sites, formation of cross bridges, formation of more link, more overlap through the power strokes, and of course, that is muscular contraction. Troponin tropomyosin complex slides back into position where it blocks the myosin binding sites of actin when the calcium is released backward and actively through the calcium pump mechanism brought back into endoplasmic reticulum. At that point, it becomes trapped and stored for the next event that will come. And finally, the last diagram that we have here is just to get a bit of idea that in a connection between spinal cord and muscle tissue, there are nerves. And we know that nerves do carry those electrically charged signals, impulses generated by our brain down into spinal cord, and then spinal cord passes them into spinal nerves, which finally reach the surface of the muscle through the neuromuscular junction, ultimately stimulating another important and interesting electrical event that leads to muscle contraction. So for the very start, or very end, better to say, at this I do have another thing that I want to add, but before we do it, I just want to close this slideshow as we won't need it any longer. To just introduce the idea that signals could be both positive and negative in both nervous system and possibly we can say in a neuromuscular junction. But predominantly it is the nervous system that we have to understand as operating on two different principles, plus minus, yes, no. To clarify it, it is like this. You have a certain idea of what to do, and you're asking your multiple friends to give you objective criticism of your plan. All of them would say, sure, sounds great, looks great, well elaborated, you can do it, go for it. So that is stimulatory for you, saying if 5 people agree that my plan is good, my chances of being wrong are simply non-existent, and you do it. So you can say that these 5 friends of yours have been good enough and they have provided support. They excited you. So that kind of feedback you received from them actually made you close to the point where you're going to start conducting action and make plan go into reality. That is exactly how multiple neurons affect another postsynaptic neuron, practically determining the outcome. If they positively stimulate, they bring its resting membrane potential closer to the threshold. That neuron received excitatory postsynaptic stimulation, EPSPS, and is more likely to fire action potential on its own. And that is exactly how you and I have been talking about action potentials all up until this point. It has been silently presumed that it's always stimulatory, that it has the excitatory nature. But how about this? This is also real life. We have some idea to do something, and we present this to a gathering of friends saying, I want to do this. And when they hear it, they jump right away and they tell you, no, it's a bad idea. Don't do that. Then you hear their opinion, you think about it for another minute, 2 or 5, and you realize if everybody's telling me don't, and I still insist on, yes, I want to do it, most likely I'm wrong, not them. So if it is some kind of idea of business plan and 5 people disagreed with it, of course, it's not going to go through. So you, as a creator of the idea, a creator of business plan, a person who presented it, actually from 5 of your stakeholders have received negative, inhibitory signals saying we don't like it. It's very unlikely to happen. And that's, again, your 5 board members are telling you no. If 5 board members are 5 neurons that are bringing negative stimulation to a neuron, making its resting membrane potential going from -70 to perhaps -80 or -90 territory, they are making it less likely that the neuron will create an action potential of its own. So that is the whole idea about in-between interaction between multiple cells. We're talking about stimulation or excitation and inhibition. It all depends which of the signals dominate. If there is more excitation, more likely it is going to result in the formation of action potential. If it is more of inhibition, it's less likely because the resting membrane potential has been pulled further away from a threshold, and action potential as such is unlikely to happen. Anyway, so I wanted this part at the very end because it could have been easily added to nervous system physiology, especially excitatory and inhibitory postsynaptic potentials. But I really wanted to talk about it when we go through synapse when we go through modified synapse, calling it neuromuscular junction, that you understand that it's not always go, 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 let's do it, but sometimes it's no go as opposed to let's go. Anyway, it is let's go to complete this presentation. It's the very end of it. I hope that it was not particularly detailed and particularly complicated to follow. I used some kind of, let's say, easygoing approach to bring this somewhat complex material within your reach, and certainly all other different details that the program you're attending requires you to know. When you have a healthy template, when you have a healthy background of information, a backbone, so to say, it is much easier to do those individual plugins and to expand your knowledge and your horizons to preferred and desired level. And I hope that you will be very successful in doing it with, I hope this is a little bit of help, what I'm doing, and that I'm not kind of wasting your time and energy in vain. For as long as you keep watching it, I will keep recording it. Thanks for your interest. Thanks for doing it. Please share it with your classmates. YouTube channel, Anatomy Made Simple by Dr. T. And I'm looking forward to seeing you in next video presentation. Until then, stay well, stay safe, and stay happy, and see you soon. Bye for now.